Hello everyone, and welcome to today's live broadcast, Signatures of Protective Natural Killer Cell Responses, presented by Michelle Poulin, PhD, and Catherine Blish, MD, PhD. We're excited to bring you this educational web seminar presented by Labroots and sponsored by Fluidigm. Fluidigm develops, manufactures, and markets life science analytical and preparatory systems for single-cell biology and production genomics. The company's systems, based on proprietary microfluidics and multi-parameter mass cytometry technology, are designed to significantly simplify experimental workflow, increase throughput, and reduce costs, while providing excellent data quality. Its products are for research use only and not for use in diagnostics procedures. To learn more about Fluidigm, please visit www.fluidigm.com. I'm Christina Jewell of Labroots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. Finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Thank you. Now I'd like to introduce Drs. Poulin and Blish. Dr. Poulin is a field application scientist at Fluidigm. She earned a PhD in immunology after completing a postdoctoral fellowship at National Jewish Health. Dr. Poulin joined the BD Biosciences as an instructor in the Customer Education Department. She joined DVS Sciences in 2012 as a field application scientist and became manager of the North American Field Application Scientist team shortly after DVS was acquired by Fluidigm in 2014. Dr. Blish is an assistant professor in the Department of Medicine and Immunology at the Stanford University School of Medicine and assistant director of the Stanford Medical Scientist Training Program. She received an MD and a PhD in immunology. Dr. Blish's current research aims to understand the successes and failures of the immune system and to better harness it to prevent infections. Her lab is perhaps best known for redefining our understanding of the diversity of human NK cells, a critical first line of defense against viruses and tumors. She has received many prestigious awards for research and mentoring and was elected as a member of the American Society for Clinical Investigation. The speaker's complete bios are found on the Labroots website. Please join me in welcoming Dr. Poulin. I will now turn the presentation over to her.
Good morning. I'm Michelle Poulin. I'm a field application scientist with Fluidigm, and I'm going to take the first few minutes of this hour to give you an introduction to mass cytometry before Dr. Blish discusses her exciting research using mass cytometry in the field of natural killer cells. As we all know, biology is inherently heterogeneous. It consists of biological systems, and biological systems consist of heterogeneous cell types, and these cell types have diverse functions and functional states. To really be able to address the complexity in this system, these systems, we need high-dimensional proteomics panels that can simultaneously address not only a breadth of cell types, but also their depth of function, all at the single cell level. Mass cytometry allows you to look at more than 50 parameters on millions of cells. The Helios, our current mass cytometry system, is our third generation mass cytometer, and it enables not only the discovery of new biology, but also the ability to do comprehensive functional profiling. Mass cytometry has been used in a range of research areas, but just for a few examples, it's currently being used in basic research as well as drug discovery and clinical research. To give you a brief overview of mass cytometry, I'd like to talk in a little bit more detail about what we call the 5 elements of mass cytometry. First are our metals. We use metals as our probes, and the reason for this is we get minimal background, which eliminates or minimizes, and in some cases eliminates signal overlap. And also, there is no endogenous cell background from these metals because they do not occur in biological systems. We currently have more than 50 high purity metal isotope tags available within our reagent catalog. With these metals, we conjugate them to our antibodies, and with those antibodies, we can build high-dimensional panels. Currently, we have more than 600 pre-conjugated antibodies in our catalog with specificities to both human and mouse, enabling both phenotyping and functional applications. These antibodies can be purchased individually or as part of panel kits that I'll talk more about in the next slide. We also sell antibody conjugation kits that allows you to label your antibody of interest with your metal of choice. And we offer 35 lanthanide metals within these conjugation kits. If you don't wish to do the antibody conjugation yourself, we also offer a custom conjugation service. In addition to antibody reagents, we also sell additional metal conjugated reagents. One example is barcoding. Others include cisplatin, which is a viability indicator, and intercalators that allow you to identify single cell events, as well as IDU, which allows assessment of S phase. In addition to individual antibodies, we also offer 18 antibody panel kits for profiling of human and mouse systems. These panels are available with up to 17 antibodies, pre-optimized, as well as all the reagents and a validated protocol for you to stain your cells. These panels can be used in combination with one another or as the backbone to create your own custom panel. As an example, I'd like to show you how we combine 2 panels with other reagents to build a high-dimensional panel. First, we can start with the human peripheral blood phenotyping kit that has 17 markers. Combine this with our human cytokine kit, which has 11 antibodies, bringing us up already to 28 markers. Then there are an additional 9 open channels where you can customize additional antibody markers. We can include barcoding, DNA percolators, S phase, viability markers, our normalization beads, as well as a few environmental monitoring channels. This brings us up to a total of 54 channels and leaves 81 open. So this means that as we expand our reagent catalog, these reagents will be able to be seen by the existing hardware of the Helios. Once the panel has been established, cells are stained with the panel. The cells are introduced into the Helios, where they are ionized in the high energy plasma. The Helios ionizes each cell and then uses time-of-flight technology to focus these isotope tags into discrete bands that are then resolved with 1 Dalton resolution by the single detector on the instrument. The Helios has a mass range of 135 channels from 75 Daltons to 209 Daltons. And as I mentioned before, this allows us to measure all our existing metal tags and any reagents that we develop in the future. Finally, we gain data from the single-cell samples. This is multi-parametric data for large number of cells per sample, and this enables deep profiling of a breadth of cell types all in a single tube. And finally, I just wanted to show the ramp in mass cytometry publications from our earliest customer to almost to the present. At this point, we're probably getting close to 160 publications using mass cytometry. And I would also note that many of these publications have been in top-tier journals, really indicating that mass cytometry enables breakthrough research. Thank you very much for your attention.
But I think I'm live, and I'll just introduce myself. I'm Catherine Blish. I'm an assistant professor in the Department of Medicine and Infectious Diseases, as well as in immunology at Stanford. And I'm going to talk today about some work we've been doing to evaluate signatures of protected natural killer cell responses. And I want to begin with just an overview as to why we're even interested in natural killer cells. And really, I'm interested in natural killer cells because they are fast-acting innate lymphocytes, and they're specialized for killing virus-infected, malignant, or stressed cells. So what we're looking at in that image is this yellow natural killer cell reaching around, sensing, and getting ready to kill that big orange tumor cell. And that orange, it can do that within minutes, and in fact move on to kill another cell. And this occurs for both virus and tumor-infected cells. In addition, they're perceived classically as a homogeneous population of cells with static functions, but recent data has really highlighted the fact that that is an oversimplification. But as a result, they've been fairly poorly characterized in humans. So we really felt that a new understanding of these cells would be more helpful. And to put this in framework, I wanted to provide a bit of an introduction as to how NK cells are specialized for killing these virus-infected and malignant cells. And really, it's important to remember that natural killer cells' function is controlled by a balance of activating and inhibitory signals, such that under homeostatic or tolerant conditions, we have a target cell. And that target cell expresses on its surface both activating ligands, which are induced under the setting of stress or infection, as well as MHC class I molecules that are found on every cell in your body. The NK cell, in turn, has activating receptors, which, of course, are specific for those activating ligands, as well as inhibitory receptors. And these inhibitory receptors are specific for the MHC class I molecules. They basically provide a back-off or an all-clear signal to tell that NK cell to bugger off and not attack, such that under normal or homeostatic conditions, that inhibitory signal wins over the activating signal, and the NK cell stays quiescent. Now, there are several activation scenarios. The first, which commonly occurs in the setting of viruses that downregulate class I or tumor cells that might reduce class I levels, is that we can have downregulation of class I and an unabated activating signal, leading to NK cell activation. Alternatively, a cell may maintain class I expression, but upregulate the number of activating receptors, leading to a dominant activating signal, and again, leading to NK cell activation. Now, the reason I introduced this framework is not to provide an immunology 101, but because there's actually an interesting implication of this model, and that is the fact that for NK cells, perhaps more than any other cell type, phenotype and function are tightly linked, because it's this array of activating and inhibitory receptors that allow it to sense signals and then transduce those signals to control NK cell activation. So to think about this another way, this also has very specific implications for how we think of diversity. And when we think about diversity within lymphocytes, we typically think about, for instance, our B cells that rearrange their VDJ to make a wide range of antibody specificities that could be specific for any virus that we might encounter. Similarly, our T cells rearrange their VDJ recombination, a T cell receptor that's exquisitely specific for a peptide MHC complex. But NK cells are different. They're really stuck with these germline-encoded activating and inhibitory receptors. So what does this mean for diversity? And really, it's a different kind of diversity in NK cells. And in fact, among NK cells, the way diversity, I would argue, is generated is that the activating and inhibitory receptors generate combinatorial single-cell phenotypes. So to draw this out a little further, this is really what we mean when we talk about diversity, is that there could be one NK cell that expresses a wide range of these receptors, another NK cell that expresses a different array, and so forth. Now, this is just, of course, one definition of diversity, but based on this definition of diversity, a few years ago, we discovered that we could define between 6,000 and 30,000 unique NK cell subsets in an individual based on this idea of phenotypic diversity. In addition, we identified, because those receptors weren't always the same in different people, more than 120,000 NK cell phenotypes in our population. And this captured a lot of attention because it was a previously unanticipated diversity of which there were both genetic and environmental influences. And I'm not going to talk too much further about that today, but I'll turn you to the paper reference at the bottom if you're interested in reading more about that. But it also raises an interesting determination as to whether there could be NK cell subsets that are tuned to be beneficial against certain pathogens. And as an infectious disease doctor, this is clearly something that I'm quite interested in. Now, another finding that's been happening in parallel around the same time is that it's been clearly and definitively proven in mice, and there's strong suggestive data in humans as well, that NK cells have memory-like features. And this is an image from a recent review in which we can see that there's NK cell memory in mouse, shown on the left side, and humans on the right. And it's been shown to both haptens, to viral antigens, specifically to murine CMV, as well as to cytokine stimulation. And similar effects have been shown in humans. So the idea that there could be this vast diversity, potentially encoding some degree of specificity, and memory, means that harnessing NK cells could provide a new approach that could be broadly beneficial in pregnancy, infectious diseases, and in cancer. So with this in mind, this really begs the question of how we should assess the function of the natural killer cell repertoire. And first, I'm going to tell a brief story about the fact that the stimulus really matters in this case. And this is a study that was actually not performed by mass cytometry, but was a study of influenza vaccination in pregnant women. I had a fellow in my laboratory, Alexander K., and he was interested in why pregnant women do so poorly with influenza infection and have high rates of vaccination. So we chose to use inactivated influenza vaccination as really a model system, as one of the few times that you can control an exposure to a virus. So we enrolled a cohort of 25 pregnant and 30 non-pregnant women who received inactivated influenza vaccination. And we drew blood at day 0 prior to vaccination, as well as at day 7 and 28 to measure their immune response. And in the pregnant women, also 6 weeks postpartum. Within these women, we assessed the NK and T cell function, and just the basic gating scheme is shown here, in which we were able to identify T cells and NK cells. And we looked at NK cell function and T cell function based on a variety of stimulation conditions, including shown here, PMA and antimycin as a positive control. And when we looked at both NK cells as well as CD4 and CD8 positive T cells, what we observed was similar to what had been published in the past. So in each case, the control women are shown in blue, the pregnant women are shown in shades of gray, and we're looking at day 0 on the left and day 7 on the right. And we observe that the pregnant women have diminished NK cell responses when we look at interferon gamma production to both in both their NK cells, CD8 and CD4 T cells, both before and after vaccination when we stimulate to PMA and antimycin. But Alex did do one thing very differently from the prior data that implies that pregnant women have suppressed immune responses. And that was that he actually looked at influenza responses in vitro. So he stimulated the PBMC cultures with live influenza vaccine, which leads to potent infection of the monocytes within the culture. So what we can see is a mock-infected culture on the left, in which there's really no viral neuraminidase staining, and 63% of the monocytes are infected on the right. And when we perform this stimulation, we observe that these infected monocytes stimulate NK and T cell responses. So on the right-hand panel now, we're looking at interferon gamma production, and we see 6.9% of the NK cells are producing interferon gamma in response to H1N1, whereas very little background and PMA and antimycin staining is higher. So now when we looked at the NK cell influenza responses in pregnancy, this was where we got our first surprise, because what we saw was not a suppressed immune response in the pregnant women during pregnancy, but in fact a trend at day 0 and a significant difference at day 7, with pregnant women actually having hyperinflammatory responses with higher frequency of NK cells producing interferon gamma. When we looked at MIP-1 beta production, what we observed was the same thing, only this was actually higher in the pregnant women both at baseline and post-vaccination. And similarly, CD107A production as a marker of cytolysis was higher in the pregnant women. So this was really quite surprising because it indicates that pregnant women have enhanced, not suppressed immune responses, but really also tells us that the immunodeficiency of pregnancy is dependent on the stimulus. When we look at non-specific stimulation with peanut animation, the pregnant women were suppressed, but in this case, they have enhanced responses to the relevant pathogen, the virus. So just to briefly summarize this short part, the increased NK and T cell inflammatory responses we believe may amplify pulmonary pathology and drive morbidity from influenza in pregnant women who actually have a hyperinflammatory responses. Antibody responses in this cohort are maintained, and in fact, the plasmablast induction might be higher in the pregnant women. But this really begs the question as to why we see enhanced immune responses in pregnancy when pregnancy has long been associated with suppressed cellular immunity. And importantly here, we looked at flu-specific responses, not responses to cytokines or PMA and automycin. Secondly, we stimulated in the context of a total peripheral blood mononuclear cell infection with monocytes, T cells, B cells, and dendritic cells all present and able to communicate with one another. And in fact, we recently published the fact that the inflammatory responses of monocytes and dendritic cells is markedly elevated in pregnant compared to non-pregnant women. And most of the data indicating that NK and T cell responses are suppressed are done on those cells in isolation, not in the context of this natural infection. So putting this together, it's possible that immunomodulatory treatments in conjunction with antivirals could improve outcomes with severe influenza. But also, and important for the data I'm going to present next, these data also highlight the importance of using physiologically relevant stimuli and also autologous infections so that we don't have to worry about MHC class 1 reactions causing false positives when we're thinking about NK cell responses. So next I wanted to really turn back in thinking about the stimulus, but now also thinking about what I told you about earlier, the NK cell diversity, and specifically what is the link between NK cell diversity and function? Does it even matter? And to do this, first we wanted to understand how stable this NK cell diversity was and try to gain better insight into what the controlling factors were. So we first performed a very simple study, which was that we talked 12 people into giving us their blood each week for 5 weeks, and then again 6 months later. And using mass cytometry, or CyTOF, we broadly profiled their NK cell repertoire over the 6-month period to determine how stable it was. And Dr. Poulin already introduced this, but the workflow for our CyTOF experiments were very similar to a conventional flow cytometry experiment in that we labeled our antibodies now, in this case with elemental isotopes. We passed them into the CyTOF machine, which has the nebulizer, then the inductively coupled plasma chamber into the quadrupole to remove the low weight ions, and then through the time of flight mass cytometer, at which point the signals are integrated and we receive our FlowJo files. In this case, this was the perfect function to look at NK cells because it allows us to look at more than 40 analytes for trillions of marker combinations. And this is important because there are at least 20 to 30 NK cell receptors we were interested in evaluating. In addition, there's really no need for compensation because of the minimal overlap. So to look at this, a graduate student in my lab, Jared Strauss-Albee, first developed an NK cell gating scheme, which is actually fairly extensive, in which we identified our cells based on DNA intercalators. We gated out the beads that we used to normalize this, then did a live cell gating, and then did a series of negative gates to avoid B cells based on CD19 staining, T cells based on CD3, monocytes based on a series of gates to get rid of CD33 positive cells, as well as CD56 negative HLA-DR positive cells, finally on lower B1 or ILT2 low cells, before finally gating on our NK cells. Within our NK cell gate at this point, we then performed, for many of these analyses, Boolean gating analyses. So the Boolean gate analyses, just at this point, we chose each marker and determined a level at which we thought it was positive or negative. And an example of many of our gates and our representative stains are shown here. And this allows us to look at each combination of receptors expressed by the NK cell. So when we applied this theory to look at the NK cell repertoires, what we found was that the NK cell repertoires are stable for 6 months in an individual. And this is just a visualization to try to capture that in a very large scale, but I recognize it's very busy. What we're looking at here is that each column with a number above it represents one individual, with each dot, the expression level of each marker coded by the size of the dot indicating the frequency of that marker being expressed on NK cells in each individual. Those that are shaded in darker are seen to be seropositive individuals, and those that are shaded in lighter stain are seen to be negative individuals. The type of marker is shown in the color, and really it's probably helpful if I blow this up. Sorry. And what we can see is that each dot represents the frequency. So if we look at this marker NKP30, what we observe is that expression level is actually quite stable between 61 and 63% at each of these time points spanning across 6 months in an individual. And we observe a similar pattern where some markers are expressed in some individuals, some markers are expressed in others, but overall within an individual these dots are about the same size, indicating relative stability. This is obviously a very qualitative way to look at this data, so we really wanted to be able to evaluate it quantitatively. And to do this, what we calculated was the 95% credible interval for the mean standard deviation of each receptor. So this is essentially the standard deviation of the standard deviations of each receptor expression profile across all these individuals for a 6-month period. And when we did this, what we observed was a range in these markers between the individuals, but relatively low numbers, as you can see here, between 0 and 0.1, with some markers that are expressed more stably at lower levels, being even lower than others. But unfortunately, this is somewhat of a unitless measure, as is obvious, because it's standard deviation of standard deviations. So to put this in perspective, what we did was we took some natural killer cells and we cultured them for 3 days in the presence of either IL-2, shown in the purple dots, or IL-15, shown in the green dots. And what we observe is that over 3 days in vitro, we observe up to 8-fold more change in these receptor expression patterns than we observe over 6 months in an individual. And keep in mind that an NK cell is estimated to live only about 14 days in vivo. So this implies there's some sort of homeostatic mechanism maintaining this NK cell repertoire. And yet they're highly susceptible to modulation. And just an example of this is shown here, where that same marker, NKP30, is shown both in the unstimulated condition and after 3 days of either IL-15 or IL-2. So from this, we really conclude that the repertoire is stable but highly sensitive to modification, which raises the idea that it could be also sensitive to optimization. And in fact, when we did this same calculation, we looked at function, and we observed that the function was similarly stable in these individuals over time. Now, the problem with what I've explained before is that's really not taking advantage of this broad mass cytometry data in which we're able to look not just at the individual receptors, but also at the combinatorial phenotypes. But when we're talking about the combinatorial phenotypes with 28 NK cell receptors, we're talking about billions of different subpopulations. So in order to come up with a better way to quantify this, we decided to borrow from ecology and use the inverse Simpson index. And this allows us to quantify the NK cell repertoire because ecologists have spent a lot of time thinking about how, for instance, you can deal with undersampling. So, which is essentially what we're doing every time we do a flow cytometry experiment. We grab about 10,000 cells and we try to extrapolate into what is happening in that individual. That's very similar to going out into the forest and counting bears. And if you go out into the forest and you count bears and you find all black bears, then there's going to be a relatively homogeneous population and a very low inverse Simpson index. On the other hand, if you count the bears and you find a few other bear species mixed in, it's still going to be a low inverse Simpson index because one species is dominating this. But there's several other species as well, whereas an evenly distributed population is going to have a very high inverse Simpson index. So what we did was we called each NK cell phenotype based on these receptor expression patterns that we determined by CyTOF as a species equivalent to a bear. And we used this Simpson index, which mathematically is inverse sum of the squares of the relative abundances of each species, to calculate this diversity. And when we looked at this, what we intriguingly found was that adults have a set point diversity. So if we look in our same 12 individuals, we observe a fairly tight clustering of each of these individuals' diversity. But what was intriguing was that some people had relatively low diversity, such as individual number 1, whereas some people had fairly high diversity, such as individual number 2. And there were some exceptions, such as individual number 10, who had a slightly broader range. But in general, when we modeled this akin to what we did before, it was a very tight range, less than 0.1 as before. So this really led us to question, what is causing this diversity? And what we intriguingly found was that the NK cell diversity correlates with the differentiation status. And we are demonstrating this here by looking at the correlation between diversity on the x-axis and CD57 expression on the y-axis. And CD57 is really a marker of an experienced or NK cell that has already gone through some sort of differentiation. And we observe a strong positive correlation between these. Similarly, we observe a negative correlation between diversity and NKG2A, generally considered a marker of immature NK cells. To drive this point further, we wanted to look in cord blood to see if this would hold true. And in fact, when we looked in cord blood, as expected, we observed relatively low expression of CD57, shown in green, whereas adults have a broad range, shown in purple. And similarly, cord blood cells in green have higher NKG2A expression. And when we looked at the diversity of cord bloods, at the NK cells that express CD57, sorry, we're having a slight connection issue, but I think I'm back online. When we looked at the NK cell populations that expressed NKG2A, so these are the NK cells that are relatively more immature in individuals, what we observed was that many of these phenotypes were shared between different individuals. In other words, my NK cells look very much like my neighbors and everyone else's, those that are immature. However, the NK cells that express CD57, those that have already differentiated in response to some sort of exposure and viral infection, are relatively more unique. And in fact, very few of them are shared among all 12 individuals. So this tells us that more unique and more diverse NK cell repertoires develop with experience. So with this framework in mind, we really wanted to now understand what implications this diversity and this immune experience has for the NK cell response to HIV. And to do this, we really needed to go back to what I was talking about earlier in terms of finding the right system to study the NK cell responses. So what we did was we developed a CyTOF panel that allowed us to capture as many activating receptors as we could, shown in blue, as well as a number of functional receptors, also shown in blue, the inhibitory receptors that basically tell the NK cell to back off, as well as a number of lineage markers. And we needed a system to really catch NK cells in the act of responding to HIV-infected cells. So to do this, we adapted systems that were developed to look at the ability of CD8 positive T cells to suppress HIV replication, and we simply used natural killer cells. And what we could show was that in vitro, natural killer cells could suppress infection of HIV in autologous CD4 positive T cells. And in fact, there's a range in the ability of individual donors to do this. So now we had a system in which we could study the NK cell response, as well as a CyTOF panel that would allow us to look in great detail at which NK cells are capable of meeting this response. And this is where the story gets a little more confusing, because no matter how we looked, we could find absolutely no predictive features that distinguished NK cells that could respond to HIV-infected cells. And this is not the case for all viruses, but it certainly was for HIV. But this led us to think about what could be determining which NK cells are responding. And what we noted was, when we turned back to this idea of differentiation, was that the differentiated NK cells were actually qualitatively and quantitatively different in the type of response they mediated. So when we looked at the CD57 positive NK cells, shown in pink, and the CD57 negative NK cells, shown in blue, what we noted was that during the response to HIV, experienced CD57 positive NK cells were significantly more likely to secrete interferon gamma or TNF, but they were less likely to kill, and they were also somewhat less likely to proliferate. There was a trend for decreased proliferation. So this implies that differentiated NK cells favor cytokine production at the cost of cytotoxicity and proliferation. But this still doesn't really tell us what is happening to the NK cell repertoire and why we weren't able to determine what is different about those interferon gamma or those killing or those TNF secreting NK cells. So we began to look more broadly at what happened to the overall NK cell population during the antiviral response. And to do this, what I'm showing here is a principal coordinate analysis. This is like a principal component analysis for those who are familiar with that, in that it's a way to reduce the dimensionality of the data and look in 2 dimensions at those components that account for the most diversity. So this is the projection of more than 11,000 Boolean phenotypes from the 12 donors we looked at, in which case each dot represents a unique phenotype of NK cells plotted against component 1 and component 2. And as we expected, we see this broad range and this diversity between these individuals because we already know that NK cells are diverse. But what's intriguing is what happens when we now expose this NK cell population to HIV-infected T cells. And although this is very hard to explain in a 2D dimension, what we're requiring now is a third component shown in color in the red to blue color coding, required to explore the same amount of diversity. And what is, I hope you can appreciate, is there's really an expansion in this cloud of natural killer cells. And in fact, you can quantify this by calculating the center of the centroid of this cloud of cells. And we can calculate the sum of the square distances from the centroid to each individual point before and after exposure to HIV. And what we observe is a significant increase in the diversity of the NK cell population during the response to HIV. So really this tells us that recognition of HIV-infected cells causes NK cells to diversify. And this potentially explains why we're unable to find a unique phenotype, because there's just so many more phenotypes. Our signal-to-noise ratio is not there. We next wondered whether this is a general feature of antiviral responses. And in fact, if we look at responses to West Nile virus infection, both before and after, we observed the same thing, this expansion in the cloud of NK cells responding to West Nile infection. So, and in fact, we can calculate this in multiple ways. If I turn back now to that inverse Simpson index, and we look at the diversity before and after exposure to HIV-infected T cells, or West Nile virus infected in cells, we observe a similar significant increase in NK cell diversity during a short-term in vitro antiviral response. So this really next led us to the question of how NK cell diversity might influence the risk of HIV acquisition. And the role of NK cells in HIV infection is quite interesting in that they're strongly implicated in pathogenesis of disease progression. However, the role of NK cells in HIV acquisition has been argued both ways. There's some very strong data indicating that intrinsic NK cell activity may be protective. And this is from an older paper from Daniel Scott Algira. And what we're looking at is the specific lysis of natural killer cells in a K562 killing assay from under EU exposed uninfected individuals. And we observed that these individuals who are highly exposed to HIV yet resist infection have much higher rates of NK cell activity than do the controls in C or seropositive people who are treated and untreated, showing other markers. And along similar lines, more recently in the Caprisa cohort, it was nicely shown that the frequency of NK cells producing interferon gamma in response to autologous HIV-infected CD4 T cells was also higher in the controls, women who did not go on to acquire HIV infection in this trial. Now, intriguingly, generalized NK cell activation is also associated with increased risk rate of infection. And in fact, within the same Caprisa cohort, if they quantified the frequency of NK cells expressing HLA-DR as an activation marker, the women who went on to become HIV-infected, the cases, had significantly higher NK cell activity than did the controls. So with this framework in mind, we wanted to study what the in vivo consequences of NK cell diversity were. And we were fortunate in that we had access to samples from the Mama Salama study. This was a study that was aimed to identify the predictors of HIV acquisition during and after pregnancy, in which we enrolled 1,304 HIV-negative pregnant women in rural Western Kenya. And this was a collaborative effort of John Kinuthia and KEMRI, the Kenyan Medical Research Institute, as well as Alison Drake and Grace Strong Stewart at the University of Washington. The incidence of HIV acquisition in this cohort was 2.31 per 100 person years. And within this sample, we were fortunate in that we had access to samples from 13 cases. These are women who went on to acquire HIV infection, as well as 23 controls. These were women who were matched for risk factors for HIV acquisition. And very importantly, we had peripheral blood mononuclear cells from matched time points pre-infection. And I stress this point because these are healthy women who do not already have HIV, but what we're trying to do is predict who is going to go on to acquire HIV based on their phenotype. And we were fortunate because we'd been doing research in Kenya for many, many years, and we knew the criteria and the risk factors for HIV acquisition, both in this cohort and other similar cohorts over the last 20 years. And thus, we were able to match our controls from among the 1,304 women based on age, knowledge of a partner's receipt of an HIV test, marital status, and history of trading sex hormone opioids. And we also observed no significant differences in the rate of sexually transmitted infections or the reporting of unprotected sex between our cases and controls. So within these, we of course did what we did, which was we performed CyTOF to look at whether we could find any markers that could predict the risk of HIV acquisition. And importantly, we looked not just at the natural killer cells, but using our CyTOF panel at the CD4 and CD8 T cells as well. And this I recognize is an incredibly busy slide, but what we're looking at is the frequency of each marker expression. The markers are shown along the bottom, and the cases in blue, and the cases in red and the controls in blue. And it's small because there's really nothing interesting here. And in fact, without even controlling for multiple comparisons, there was no single marker that could predict the risk of HIV acquisition. And this includes the generalized immune activation status of T cells, which has been associated with disease progression and thought to perhaps contribute to risk. But in this particular study, it did not do so. However, we got a surprise when we went back to our inverse Simpson index and we quantified the NK cell diversity. And what we found was that there was a significant increase in NK cell diversity among the women who went on to acquire HIV infection. And that's displayed here, and you can see the shift in the curve. And in fact, we can quantify this risk. Obviously, there's significant overlap. But nonetheless, based on a logistic regression, the odds ratio for HIV acquisition were 2.5 for every 100-point increase in diversity. So just to put this in perspective, what we're observing here is an approximately 500-point range range in the scale of NK cell diversity. And for every 100 points on that scale we increase, the risk of acquiring HIV is 2.5-fold higher. The risk of acquiring HIV associated with, for instance, ulcerative sexually transmitted infections such as syphilis is about 3 to 4-fold. So this is actually quite a substantial risk. In addition, the reduced risk in male circumcision is similarly about 2 to 3-fold. Both treatment of syndromic STDs as well as circumcision were thought to be significant enough risks to launch multiple randomized controlled trials to try and prevent HIV acquisition. In fact, it was also only the diversity of NK cell receptors that predicted HIV acquisition in this study. So really, to put this together, what we've managed to build with our high-profile analysis is a model of NK cell diversity. And this is only a model that requires testing, but I put this forward to drive further research. And what we believe is happening is that we all start out with a naive baseline NK cell repertoire that's actually relatively homogeneous. And as we go through life, we're exposed to different viruses, and each of these viral exposures leads to diversification and specialization of the NK cell repertoire. And each subsequent exposure is going to further diversify and specialize that repertoire. But what's intriguing to think is that this might have its limits. And what we don't know is, does this diversification and specialization lead to a decreased ability to respond to a de novo pathogen? Now, importantly, this does not eliminate the possibility of NK cell memory, because while we might be less good at responding to the red virus after seeing the purple and the green, we might be, in fact, better at responding to the green virus the next time we encounter it. So to bring this to a conclusion, what I hope I've convinced you of is that the human natural killer cell repertoire is astoundingly diverse. The diverse NK cell repertoires are associated with terminal differentiation and cytokine production, perhaps at the cost of killing activity and proliferation. That short-term exposure to virus-infected cells augments NK cell diversity. And that in a cohort of Kenyan women, NK cell diversity predicts the risk of HIV acquisition. But of course, confirmatory cohorts are needed. This was a very small study, and efforts are ongoing in that regard. But what's intriguing to think about is that this tells us that NK cell diversity may in fact not be beneficial to the innate immune system. And this is a somewhat heretical thought, given that we always think of diversity as such a great thing in the immune system. But of course, it's important to note that it could just be a correlative risk, that there could be some unmeasured factor that's correlating with the NK cell diversity, and that factor is actually what's driving this association. Interestingly enough, regardless of whether the NK cell diversity is a correlate or is actually playing a causative role in this increased risk, assessing the prior experience of the innate immune system could have both functional and clinical utility in predicting the vigor of its response. So to finally end, I just want to acknowledge the tremendous efforts of my lab pictured here, and in particular Dara Strauss-Albee, who was a graduate student who led all the HIV studies, as well as worked closely with Ruth Montgomery and Yi Yao at Yale on the West Nile studies, as well as a number of other investigators in my lab, Olivia Hatton, who has worked on EBV and some work I didn't have the chance to describe, Lisa Kronstadt working on influenza, as well as Alex Kay, Emily Wang, and Nicholas Bayless's work on influenza during pregnancy. We initially developed the CyTOF panels in collaboration with Peter Parham and Amir Horowitz at Stanford, and had significant help from the Nolan Lab and the Davis Lab, as well as Holden Maker and Michael Eipold at the HIMC. And we're indebted to our amazing statisticians, Susan Holmes and Julia Yukiyama, who have helped us deal with this highly complex data. Thank you very much.
Thank you very much, Catherine, for that exciting and informative presentation. At this point, we're going to move into the question and answer session of the seminar. So you can submit questions and answers by clicking on the green Q&A button at the lower left of the presentation window. Type in your question in the box that appears on the screen, and then click send. And we're going to try to answer as many of your questions as we can within the time allotted. So just to begin, there were a few questions that I'll begin with based on my short portion of the talk. The first one was, did you say heterogeneity at the single cell level? Just to clarify, what my meaning was while talking in the first couple of slides is that because of the complexity of biological systems, we need to be able to address that heterogeneity. Mass cytometry enables you to do that by being able to look at very high dimensional panels, which allows you to analyze both the breadth of cell types in your system of interest as well as their a depth of function. And this is gained by being able to look at many, many markers simultaneously and get that data at the single cell level. The second question was, can we identify the population based on the cell dividing status at the single cell level? And Catherine may also want to discuss this, but we do, from Fluidigm, have reagents that allow you to address cell cycle. We have iododeoxyuridine to be able to look at S phase, and then by utilizing antibodies against certain cell cycle phase specific markers, we're able to assess all ranges of all phases of the cell cycle. The next question will be for Catherine. And the question is, could you address a bit more on why pregnant women with increased NK response have poor cellular immunity? Is this mean that pregnancy is bad for cancer treatment?
Okay, I'm going to take that over. Well, there are a few things to keep in mind. My point was really not that pregnancy is neither good nor bad, and that it's a complex state and it depends on what specific function you're looking at. And I think there's a misunderstanding and a massive oversimplification that gets repeated over and over, that pregnancy represents an immunodeficiency state. And that makes some logical sense because pregnant women have to tolerate their semi-allogeneic fetus. But I think it's important to keep in mind that not all arms of the immune system are necessarily suppressed and not all responses are suppressed. And in fact, there is depressed cellular immune responses if you look at a number of different factors, including cytokine stimulation and, as I said, responses in isolation. But in fact, the innate immune system is activated potentially to make up for some of that suppression. And it's been well described that there are increased regulatory T cells and a number of other factors going on during pregnancy. In terms of the setting of cancer treatment during pregnancy, that is a whole ball of wax, which is honestly difficult to address because most of the agents used to treat cancer are potentially harmful to the fetus. So it really is on a case-by-case basis whether any immunomodulatory treatment of cancer is going to happen. To my knowledge, there's no data indicating that pregnant women are more likely to develop cancer during pregnancy based on this depressed cellular, some aspects of cellular immunity being suppressed during pregnancy. And I guess the final point about that is it's important to note that many autoimmune diseases get better during pregnancy based on the types of immune responses that are suppressed. Though that's not universally the case. There are a few, such as lupus, typically antibody-associated diseases that can actually undergo flares during pregnancy, but the vast majority improve significantly during pregnancy.
Thank you, Dr. Blish. The next question is also for you. The question is in two parts. When investigating diversity, A, how do you, and is it relevant to distinguish functional versus lineage defining markers? And B, do you subset this data set to track specific subgroups over time?
Okay, just waiting to get it. So the first part of that question was, how do we distinguish diversity? And that is a very good question, and I didn't go into great detail on that. And it really depends on what the question is. For most of the data I just presented, we referred to phenotypic diversity in terms of the array of activating and inhibitory receptors. And our interest was whether, if we looked at functional cells or non-functional cells, there were any particular patterns of these receptors on them. So in that case, we were looking just at the phenotypic markers in terms of diversity. So you'll end up with a different level if you look at functional diversity as well. And in fact, we analyzed the data both ways and many other ways that could take another several hours, if not days, to describe. But there's a huge amount of functional and phenotypic diversity. It's also important to note that we do see both heterogeneity in terms of phenotype and function, but it's so great in HIV, unlike many other viruses, that we're unable to distinguish specific phenotypes. In terms of specific subsetting and following up the individuals, I'm not sure I fully understand the question, but our goal was to identify specific NK cell subsets that we thought were particularly responsive to a given virus, and then, of course, be able to sort them and discover whether those were, in fact, uniquely able to respond. And in fact, in HIV, since we weren't able to identify a phenotypic subset, we were unable to do that. In terms of looking at our cohorts and looking at specific outcomes within the cohorts, unfortunately, these are too small cohorts. We simply don't have the power statistically to do any further subsetting, but we're looking to acquire additional cohorts and answer some of these same questions in a confirmatory independent cohort, which is actually much more statistically robust.
The next question is also for Dr. Blish. Did you study HLA subtypes to stratify NK cell diversity in response to infection? Does HLA diversity impact the NK cell repertoire diversity?
Great. That's an excellent question. So we did HLA subtype everyone, Again, these were fairly small studies, so our power was fairly limited. But I turn people to the first 2013 paper, we describe it there. We were actually surprised that we did not see an obvious significant association between the NK cell repertoire and the HLA subtypes in terms of education. In other words, we didn't see a significant imprint, phenotypic imprint of education on the NK cell repertoire. Within the African cohorts, frankly, the HLA diversity is so great that within a cohort of about 36 people, we were really unable to subset, again, based on different HLA types to see if there was a specific association with disease risk. But overall, it appears as though the NK cell repertoire is formed somewhat genetically in terms of the array of KIR, NKG2A, and LoRBR1 with an overlying diversity of the activating receptors. But that the impact of that, the HLA educating those cells through KIR or HLA-E, NKG2A, is not noted in the diversity as we measure it phenotypically. But that may require just hundreds and hundreds more.
The next question is also for Dr. Blish. When you define a subset in a Boolean way, do you include functional markers?
Again, the question was about Boolean gating. Again, that depends on what question we're asking. We, in fact, looked both at median expression and at Boolean gating in terms of determining the frequency of functional NK cells. But we did not define our diversity based on functional markers in this setting, though if we did, it didn't really change any of our overall conclusions. We analyzed the data both ways, but I did not present that data.
Next question for Dr. Blish. What maintains the repertoire diversity within a person? And how come newly developed NK cells are not more homogeneous? Bone marrow transplants, does stroma microenvironment affect person-specific diversity?
Okay, there were about 3 questions in there, and I'm hoping that I got them all. So the first is what maintains the NK cell receptor diversity, and I presume that means in adults. And the short answer is we honestly have no idea, but it's an active area of investigation. The long answer is that we have some hints, and those hints come from both our work and, importantly, many other investigators. So first of all, and agreeing to that, there is relative homogeneity of the developing NK cells, but there's also a very regulated pathway of NK cell development. Typically, they express NKG2A first, then a subset, depending on the HLA haplotype, will downregulate NKG2A and upregulate an array of KIRs, and that has a huge impact on the diversity. And the extent to which that's influenced by the bone marrow stroma is not known, but it is known that education through HLA molecules have a significant impact on that. In terms of once the NK cell has gone through that developmental pathway, and it's either an NKG2A or a KIR or even a null expressing NK cell, how that diversity is maintained remains unknown, but there's definitely hints that there could be some epigenetic modification. And this comes primarily from work looking at CMV infection, which causes an imprinting on the repertoire. And the NKG2C expressing NK cells have a significant epigenetic imprint that may play a role in their diversity. And I see we're running low on time, so I'm going to stop there.
Thank you. We have time for a few more questions. The next one is, again, for Dr. Blish. Can you comment about the potential NK memory subsets? Do you think these can be found in HIV-infected individuals?
So I started this project hoping that we could find NK memory subsets in HIV-infected individuals, and I mean, the truthful answer is I don't know. That said, there's very strong evidence that the NKG2C, CD57 positive NK cells are CMV memory-like cells. And we can argue all day over what you call a memory cell versus not. But in addition, I'd point folks to Keith Reeves' work in the macaque system, in which they've identified NK memory cells for SIV. And so I think it's not impossible, but I think that there are really two possibilities. One is that HIV has come on the human population so recently that honestly the NK cell response has not evolved a memory population to deal with a virus like HIV. And the other is that we just need to keep searching, and it's more subtle. And so efforts are ongoing.
Great. The next question for Dr. Blish. Do you suspect that NK cells become exhausted due to ongoing exposure to virus?
It's a very good question. And I suspect yes, but I'm not sure if it's going to be in the same way that T cells become exhausted. And that's something we'll explore. Certainly there are a subset of NK cells that express markers such as PD-1. In addition, there's a lot of really exciting data from other groups about TIGIT regulating NK cell function. So I think it's not impossible to think that NK cells could undergo a similar degree of exhaustion. And then in fact, some of those immunomodulatory drugs like PD-L1 and so forth could be acting on NK cells as well as T cells.
We're almost out of time, so this will be the last question. For Dr. Blish, do you have a model as to how the NK cell population would accumulate diversity over time and experience?
So the question was, do I have a model as to how the NK cell population would accumulate diversity? And I don't have a model beyond what I displayed, but what we do find is that exposure during an acute response to a virus, at least in vitro, we observe an increase in diversity. And presumably, for this model to hold, some of that diversity would be maintained and that we would see similar effects in vivo. We do not have the in vivo data yet to prove that, so it is just a model, but one we're actively exploring.
Thank you very much, Dr. Blish. I'd like to thank everyone who has attended this session today and for your thoughtful questions and participation. I'd also like to let everyone know that today's webcast will be available for on-demand viewing through March 28th of 2017, and you will receive an email from Labroots letting you know when this webcast will be available. Feel free to share this announcement. So that's all for now. Thank you very much for joining us, and we hope to see you at future Labroots sessions. Goodbye.